Okay, so it is a great pleasure to welcome our annual Picower lecture speaker, Eve Marder. Eve is the Victor and Gwendolyn Bainfield Professor of Neuroscience at Brandeis University, where she is also the head of the Division of Science. Eve received her PhD in 1974 from UCSD and conducted a one-year postdoc at the University of Oregon, and then a 3-year postdoc at the École Normale Supérieure in Paris. She became an assistant professor in the biology department at Brandeis in 1978 and was promoted to professor in 1990. During her time at Brandeis, it was instrumental in the establishment of the undergraduate and PhD programs in neuroscience. So over the years, Eve has made seminal contributions in systems neuroscience and really fundamentally changed the way neuroscientists think about how nervous systems work. So first, using the STG circuit, which generates rhythmic behavior, in the quotations, Eve showed that the STG can produce a wide range of outputs modulated by actions of several neuromodulatory substances. So her work demonstrated for the first time that circuits are prone to reconfiguration and no network is truly hardwired, an assumption prior to her work. And Eve further used both the computational models and experimental studies to explain how a stable network can be maintained throughout the lifetime of an animal with such high degree of modulation. This led to the discoveries concerning the mechanisms of neuronal compensation that allows the incorporation of plastic change in biological networks without destroying their overall function. This is also known as homeostatic plasticity, and Eve is truly pioneer in this area. And I think homeostatic plasticity not only widely applies to neuroscience today, but it's really, I think, all how the biological systems work. And really thanks to Eve's seminal discoveries. And I think most importantly, Eve's lifetime work really helped us interpret and comprehend the most sophisticated function of mammalian brains, especially human brain, and how compensation and homeostatic plasticity may maintain the cognitive function in aging and other neurological disorders. And throughout her contributions in science, Eve has received numerous awards, and I think it increases by the month. And she is also a member of the National Academy of Sciences. And today, I think we are extremely fortunate to have Eve here out of her extremely busy schedule. And I think she changed the title of her talk a little bit, but please let us welcome Eve.
You can unmute. It's a great pleasure to be here, and a great privilege to see so many old friends. Actually, some friends who are older, and some friends I've had for a long time, and anyway, and some new young people. And I just want to say I had a fabulous day, including meeting some of the most recent assets or additions to the MIT faculty. So what I want to do today is sort of a hybrid that will try and sort of capture for you some of the lessons learned from, as it says now, 40 years of working on a small nervous system. Now, is there something, can you hear me? Sorry. Yes. So, now can you hear me?
Yes.
I'm not going to start all over again. I didn't say anything. Okay. But what I do want to do is try and capture for you the lessons learned from the last sort of 40 or 45 years of working on small nervous systems as we try and understand how circuits produce the dynamics that are important for behavior. Now, some of you know that I've worked on the crustacean stomatogastric nervous system my entire scientific career. And the stomatogastric ganglion, as you'll see in a moment, has only 30 neurons. So you might ask, how is it possible to have thousands of person years, it's not maybe thousands of person years, I have to do that calculation at some point, devoted to the dynamics of 30 neurons. But I think what it is fair to say is over the years we've learned many, many, many lessons which are general to understanding of vertebrate nervous systems and human nervous systems. And I'm going to try and give you sort of a sense of what those questions and answers are and how we've been able to use a small, very well-defined circuit to extract secrets and general principles from. Now, before I start, I'd just like to thank the people in my lab who've done this work. Why is this funny? Okay, first of all, my present lab is up here. The people whose name in green are the people who have contributed most to today's talk. For those of you who are in purgatory, that's graduate school postdoc, these are all the people who went through my lab as graduate students and postdocs who now have independent positions, who are doing science in their own right. And I just feel that it's just been an extraordinary joy to have so many wonderful people with whom to work. And I'll try and mention their names as I go. So the basic problem that I'm going to be talking about in the second half of my talk, and then I'll be sort of setting up with the first half, is the problem of building brains to work in the world. What really matters is, would some of you like to come in and sit in these empty seats so I don't have to look at people standing because it makes me very nervous and anxious? You'd think after all these years that nothing would make me nervous and anxious, but people standing during talks makes me nervous. And that's because I hate standing, so thank you very much. So we know that we have to build brains to work in the world. And the key here is that we as biologists, and I'm going to come back to this, have to confront a problem that is sometimes I sort of start by telling an audience that we as biologists sort of have chosen to forget something that we knew when we were 2 years old. And when we were 2 years old, we knew that all brains are different as a consequence of differential history and genetics. And as experimental biologists, we chose, especially neuroscientists, we chose to forget this and to pretend that all mice are the same or all cats are the same, and that when you calculate means and standard errors, that those means and standard errors actually capture what every individual would have for that parameter. And one of the lessons I'm going to sort of weave into this is that we have to go beyond the mean and go back to really embracing the actual differences in parameter combinations if we're actually going to understand how brains really work. But I think you'll see that as it comes back. And so the underlying sort of principle that my lab is really trying to grapple with right now is how tightly tuned, or question, do the parameters that govern synaptic strength and intrinsic properties need to be for good enough circuit behavior? Or how different are the underlying structures of normal healthy brains? Now, obviously, if you want to understand the boundaries between health and disease, as so many of you in the room care to, if you don't know what the range of any given parameter in the normal healthy population is consistent with good enough behavior, it's very hard to know where those boundaries between health and disease are going to come from. And so the tolerances of that are extremely important. And just to sort of keep sort of belaboring this for one moment, I often in these audiences say, I can tell you with 100% certainty that all of you have respiratory centers that are good enough. And that's because you're here and you're breathing. And I am certain that nobody in the room thinks that you have identical respiratory centers. So once we start there, then we can ask, well, how different really would those respiratory centers be? How many of you have how many pre-Botzinger neurons, and what's the density of the inhibitory and the excitatory connections that are important for your respiratory rhythms? And So one of the things that we and some others have been doing in the last 5 or 6 years is really starting to try and put numbers on answers to questions like that. And that will be really what I'll be talking about in the whole second half of my talk. I'm going to start with talking, just briefly introducing to you the pyloric rhythm of the stomatogastric nervous system, because this is the canonical sort of one of the 2 best understood small nervous system or small circuits. And I think it has a number of general principles to offer. So what we do is we go to the fishermen and we buy lobsters and crabs from the fishermen. And therefore, we're studying a wild type population of animals that have all been good enough to live in the North Atlantic, in the Boston Harbor, right, for 5 to 7 years before we get them. So I want you to remember this. These are all adult animals that have lived in that Atlantic Ocean for 5 to 7 years. So they're all good enough. I mean, good enough to live in the Boston Harbor, which is better than you and I could do, but that's another part of the problem. What? You can live in Boston? Was that us? Okay. A hiss? Okay. Then what I used to do, and my graduate students and postdocs and undergraduates now do, is we take the stomach out of the lobster, or the crab. Most of the rest of the animal goes into the freezer, and other things happen later. Not me, I don't eat lobsters and crabs anymore, but they do. Anyway, and then we dissect the nervous system off the surface of the stomach and pin it down in a Silgard-lined petri dish. This is a full-size petri dish. And what you see in this preparation are 4 ganglia. These 3 ganglia include neurons that send descending modulatory projections into the neuropil of the stomatogastric ganglion. Here's the stomatogastric ganglion. It has 26 to 30 neurons, depending on species. These are large neurons. They have 100 micron somata, and therefore it's very easy to do intracellular recordings. We simultaneously record 3, 4, sometimes 5 with electrodes at a time, so we can record intracellularly from 3 or 4 neurons at the same time if we want, or we can put 2 electrodes in some cells. We also record extracellularly from motor nerves, and therefore can monitor the performance or the behavior, the output of as many as 30 neurons at a time simultaneously. And we have miles and miles and miles of recordings. And we, the community, have had these miles of recordings since the mid-70s. And so one of the things that's very interesting, I sometimes get very amused when people think that just having simultaneous recordings from many neurons will all of a sudden solve your problems. It just means that you now have to develop new techniques to analyzing those data. Anyway, so we record. What we see is we see a triphasic motor pattern, much like your respiratory motor pattern. The pyloric rhythm governs a valve at the back end of the animal's stomach. The LP is a constrictor motor neuron. PY is another constrictor motor neuron, and PD is a dilator motor neuron. So what we see is LP, PY, PD, LP, PY, PD in these simultaneous intracellular recordings. And in the extracellular recordings, you again see LP, PY, PD, LP, PY, PD. Now, most of the neurons in the ganglion are motor neurons, but the connectivity is at the level of the motor neurons, and this has facilitated the analysis of the connectivity that gives rise to these motor patterns. Because with one set of recordings, we can both see the motor pattern and the circuitry that gives rise to that motor pattern. Now, this used to be called a connectivity diagram. Today it's called a connectome. I will flip and use both terms. What it is it's a wiring diagram for the connections of these neurons. And what we see is a single neuron. It's 1 neuron per ganglion, called the AB neuron. It's an interneuron, and it's a bursting pacemaker neuron. It's electrically coupled to the 2 PD neurons. And then all of these chemical synapses are inhibitory. So what we see is the rhythm exists because of the pacemaker properties of the AB and the PD pacemaker kernel, the 3-cell pacemaker kernel. The pacemaker kernel rhythmically inhibits the follower cells, which then fire on rebound. So we have a rhythm, and then the phase relationships, or the rest of the firing, is set by the strength and duration of these inhibitory synaptic connections, and the intrinsic membrane properties of the follower cells, which contribute to the rebound. Okay? And the LP rebounds before the PY. So what happens is the pacemaker bursts, and then it shuts off LP and PY. LP rebounds first because of actually the potassium currents and in LP and PY. Then PY eventually rebounds, shuts off LP, and the whole cycle continues. Now, if I had just shown you this and not shown you this, there's absolutely no way you would have been able to go from this connectivity diagram to predict these dynamics. And that's because, and Corey Bargman working in C. elegans will say the same thing, a connectivity diagram is absolutely necessary and completely insufficient to help you understand how a circuit works. You can't understand how a circuit works without a connectivity diagram, but it really is only the beginning rather than the end. What you would need to go from a connectivity diagram to dynamics is you'd need all of the voltage and time dependent currents in each of these cells. You'd need to know the strength, time course, and time dependence of those synapses. And then you'd need some way of putting all that information back together again. And what that routinely would be building some sort of computational model that would allow you to rebuild the system on the basis of its component parts. And that's partly why almost 20 years ago, or more than 20 years ago, I started collaborating with theorists because as I was trying to go from understanding circuits to understanding dynamics, it was clear I needed to have some other sort of computational tool to basically to try and get back to dynamics. But I think that's lesson number one. A connectome is necessary and completely insufficient. Now, you need it. You absolutely need it. But it's just the starting place. Now I'm skipping 35 years of work and summarizing it in this one slide. This is what we know about the neuromodulators involved in the sympathetic gastric nervous system. 2012, probably we've added something, but it doesn't matter. When I started studying neuromodulation, the earliest work we had done was on dopamine and serotonin and a couple of other amines. And then we started studying a peptide called proctolin. And after we had identified the proctolin pathways, and I kept on looking for more modulators, someone said to me, well, you have dopamine and proctolin, why don't you just stop? You know, the next one isn't going to be interesting. You know, enough modulation is enough. And I said, if I'm going to understand neuromodulation in the context of this circuit, this nervous system, I need to know all the cast of characters, because I'm not going to be able to understand the whole structure of the neuromodulatory control systems without all the players. So over the many years, we've been working on getting all those players, and this is probably 80% complete. And it was very hard work in the early days. It's much easier now with modern mass spec methods. But basically, the answer is it's not one. It's not dopamine, acetylcholine, and serotonin in your brain. And it's not one peptide. It's many. And the answer that it's many is probably true of every brain region, probably some more than others. But I'm sure that this image is more true than not everywhere you guys work. What you see there are 25 pairs of descending modulatory neurons, and they have, as co-transmitters, and all of these substances are used as co-transmitters, acetylcholine, GABA, nitric oxide, dopamine, histamine, octopamine, and then a whole ton of neuropeptides. Most of those substances are also released into the hemolymph by neurosecretory structures. The stomatogastric ganglion sits in an artery, so it gets both hormonal release and local neuromodulatory release. So one of the other lessons that we see with this that we would not have known otherwise is the same substances may be setting a low concentration baseline neuromodulatory tone, and they may be used as co-transmitters in descending modulatory neurons that are activated by defined sensory inputs that basically then are reconfiguring the network rapidly under certain behaviorally significant situations. So that's a very different sort of way of thinking about neuromodulation than we were thinking at the beginning of all of this. But I think this really is an image that you should hold on to because, and I think this image is basically what the C. elegans people are starting to see and that we're going to see in Drosophila and we're going to see everywhere. Okay, now what do modulators do? And you all know this. This is a very old slide. Came from the thesis work of Judith Eisen. It was published in '87. She did the work in the early '80s. And she isolated the AB cell and completely washed the preparation, removed the PD cells, removed all the modulators, etc. And under those conditions, the AB cell was silent. And then here she applied pilocarpine. That's a muscarinic agonist. And you can see the oscillation comes back. And now this was done with a chart recorder. So it was a slow time base, and then she pushed the button, and now the chart recorder went faster, and so now you can see those oscillations spread out, and you can see these very beautiful high amplitude, number of spikes per burst oscillations. And then she did the same experiment with serotonin, and again here, serotonin turned on the burst. Something must have happened here with the trace in the chart recorder. We don't want to talk about it. And again, you can see the complete transition from off to slowly growing amplitude and frequency of the oscillator to a very robust oscillator. So this is an example of a modulator that completely qualitatively changes the intrinsic properties of a neuron in a circuit. And this happens to be our pacemaker, so this is important. These are more recent data, but using a peptide called RPCH, that's reg pigment concentrating hormone, it doesn't matter. There's a synapse, these are intracellular recordings, a synapse from LP to PD, which in control saline is virtually invisible. This was from the Maine lobster. And then in the presence of RPCH, you see this giant IPSP. So this is a case of an anatomical synapse which requires neuromodulator for the expression of the physiological correlate of an anatomical synapse, which is there. So the anatomical synapse is the backbone of that connectome, and then the modulatory environment creates the tone, which now produces the functional synapse. Now, take home lesson number 2 or 3 of the work on modulators, and this comes from a lot of work that my lab did and that Ron Harris-Work's lab did over many years. If this is the full circuit for the pyloric rhythm, every single neuron in the circuit has multiple currents that are subject to modulation by many substances. So if each neuron has 10 or 12 voltage-dependent conductances, many of them can be modulated by one or another substance. Sometimes, as the case for serotonin in the LP cell, serotonin will modulate 3 or 4 different currents in the same cell. Sometimes, as for many of the peptides, they converge to act on a single current, depending on where it is. But in any case, you have that. And every synapse is subject to modulation by one or more substances. So you have a backbound circuit, and every parameter is, in a sense, free to be tuned by neuromodulation. So when we first started studying neuromodulation, we said, oh, this means you've got all this potential for flexibility. And that's what I'm going to show you in the next slide. These are old experiments that Jim Wyman did many years ago. And again, he took an STG, he washed it extensively, removed all the modulatory inputs, and then applied pilocarpine, he washed, applied serotonin, washed, proctolin, washed, dopamine, etc. And you can see each one of these substances produces a different characteristic and different change in the dynamics of the motor patterns. And at the time, we said, this is a mechanism for motor pattern flexibility, and it is that. But over the years, we realized something else which is more important, or as important. First of all, any number of substances can reactivate the motor pattern. And they all do slightly different, they do so slightly differently, but any number of one of them can serve that function. So if it's important for it to be on, it can be on using a whole lot of degenerate mechanisms. Now, that's number one. Number two, of these substances, the only one which at high concentrations really produces what I would call a crash is here, dopamine, where you lose the fundamental alternation between LP and PD. They are functional antagonists. It's very bad. The circuit can't work if they're not firing an alternation. So dopamine produces a crash, but all these other substances produce different forms of the motor pattern, but basically you still maintain the LP-PD alternation. So today, we think about this a little bit differently. First of all, we'd say that circuit reconfiguration exists, and the temporal dynamics and modulatory environment construct a functional circuit by tuning functional synaptic strength and modulating neuronal excitability. That is certainly true. What it means is that you have the connectome, or the anatomical circuit, and then you create a functional circuit with all these neuromodulatory substances and the activation in the circuit. And so the functional circuit that exists during a given behavior is that set of solutions, that set of parameters, and not all the other potential ones that could be there. And that is true. Now, the problem if you're an electrophysiologist is that means you would want to be able to measure the intrinsic membrane properties and the synaptic strengths while the circuit is working, not the way we would routinely have done it 25 years ago or today in the lab, which was by silencing the circuit so that you could actually study a pair of neurons or the voltage clamp a neuron in question. And we'll come back to that. So, but the important other sort of question raised by this, and this is very relevant to the way we think about mental illness and seizures in the vertebrate brain, is the neuromodulatory conundrum. How can circuits be reliably modulated, as they are, without running the risk of overmodulation? If every single parameter in a circuit is in principle modulatable, any of you who've ever played with a computational model of a circuit knows that you do anything and the thing crashes, right? It's much easier to crash a model of anything than it is to find the places where it's stable. And now we have modulators in God knows what combinations and in God knows what concentrations, and the circuits rarely crash. Your circuits in the brain, the circuits in our population, don't crash very often. Schizophrenics are, 1% of the population. 1% of the population is seizures. And yet we've got all these modulators floating around, released, that are capable of, if you were building a model, of crashing things much more easily. So I think there really is a deep set of questions about how neuromodulatory control can be as widespread and as significant without running into the risk of crashing circuits. And this is one of the things that I have to solve before I give up science. I've been working on this for a long time, but I'm getting closer. Okay. Now, I've just given you the first part of the STG motor patterns, which is the pyloric rhythm. But that's an oversimplification. There's actually a second rhythm. And the second rhythm is the gastric mill rhythm. So on this time base, which somehow or other got lost from the slide, the pyloric rhythm is here, LP-PY-PD, and it's fast. It has a period of about 1 second. But you also see a second rhythm, the gastric mill rhythm, which you can see here as a depolarization of LG, alternating with DG, LG, DG, LG, wherever it is, DG. And so you can see a fast rhythm and a slow rhythm. And the important thing is also that there are these two other neurons here, you see, IC and VD, that are firing in episodes in time with the slow rhythm, but they also are firing in phase with the fast rhythm. So these neurons are multiplexing a fast and slow rhythm. Okay, so this is what the STG really does. And the gastric mill rhythm moves teeth in the stomach, and the food goes chewed by the teeth, and then after it's been chewed, it goes into the filter, and that's the pyloric rhythm. Okay, this is the real, I don't know how real it is. This is the full connectome of the STG with some of those neurons I was showing you before over here, and now these gastric mill rhythm neurons. Now, first of all, it's not like they're two circuits and they're anatomically separated. These guys are all intertwined into the same neuropil, much like a lot of the circuits in your brain. And notice all these electrical synapses. And notice there are electrical synapses between those neurons in the fast circuit and the slow circuit, and they cross over. And the real nightmare, and this is, as far as I'm concerned, one of the hardest problems of studying any circuit, any real circuit, is what I call parallel pathways and the confounds that come with electrical coupling. So if you just look here, LP is electric coupled to IC. There's a reciprocal inhibition between IC and PD here. There's also reciprocal inhibition between LP and PD over here. So IC can influence PD polysynaptically through this electrical junction or monosynaptically here. And likewise, there's a similar circuit over here and other places as well. So whenever you have parallel pathways and electrical junctions make parallel pathways, that's their job in life as far as I'm concerned, is to make your life totally miserable for the electrophysiologist. People think that they have to do with synchrony and all that. I don't think so. Their job is to make understanding circuits really hard, okay? Because that's what they do. Anyway, you know. And notice, in this circuit, as will certainly be true once the vertebrate world catches up with us, there are many electrical junctions between neurons of different types, right? You know, okay, there are two PD cells are very tightly electrically coupled, but the LP and IC are very different neurons. And IC and LG, all of these cells are different, and they are also electrically coupled. Okay, so a few years ago, a year ago, we were thinking about, well, wouldn't it be nice if we really wanted to understand how LP, for example, influences everybody else in the circuit? Wouldn't it be nice to have a functional measure of how information is really traveling through this sort of network? So we started looking around through all the vertebrate literature, trying to see if there was any way of getting what we can't get. What we would want is, when the circuit is doing X, whatever X is, we would like a measure of all the synaptic strengths, electrical and chemical, during the circuit's operation. And of course, we'd also like to know what all the intrinsic properties of the neurons would be. So we started looking for ways into that problem. And we just sort of said, okay, well, but these smart people working in cognitive neuroscience use all sorts of measures for producing functional connectivity. So maybe we could use some of those for functional synaptic strength. And Granger causality is one of the ones that's used for functional connectomics or connectivity in the higher level people. So we thought, well, we would give it a try. So first of all, we discovered that most people thought that Granger causality, which just is a way of predicting whether activity, basically saying that activity in a putative presynaptic neuron predicts activity in a follower neuron, or set of circuit, whatever. Most people thought it couldn't be used for inhibition, but it turns out it works fine for inhibition. Remember, ours is an inhibitory circuit, and we validated that it works totally fine with inhibition. And Tilman Kaspersky, who was a postdoc who did this work, just built very simple model neurons, hooked them up with defined connectivity, first in a cyclical network architecture, and then in a pyloric-like network architecture with all these inhibitory connections, and then ran the Granger algorithms. And in this circuit, where each of the neurons was basically an integrate and fire type neuron, the Granger causality analysis reported back basically the correct connectivity. So, okay, but now we tried to use it with real STG data. And this is pyloric rhythm data that Gabriel Gutierrez had collected. There's 4 different animals. So one set of recordings, a second, third, fourth set of recordings. And this is the right answer. So they ran Granger on each of these 4 sets of recordings. And what Granger almost invariably, not in every case, but almost invariably did, it returned a connection from PY to PD, which is not there. And it did that routinely. And it made some other things it got right, but it got that most notably wrong. And then we saw this, and then we thought for 1 second, and we realized what the answer was. The answer is the PD is a strong oscillator. It always fires after PY. Therefore, the algorithm thought there was a functional connection between PY and PD because PD always fires after PY, but PD always fires after PY because PD is a strong oscillator. And then we realized that the minute you have strong or even weak oscillatory dynamics in the circuit, the Granger predictions will return a false causality, or a potentially false causality. And this isn't surprising, and so now there are people trying to come up with different kinds of algorithms, etc. But I stepped back and said, my God, all these people who are using Granger and like algorithms for connectomics at the level of networks where you don't know the right answer might easily be fooled by the brain rhythms that we know are all present. So at some level, this is a trivial result, because it's obvious why it gets it wrong. And at another level, it turns out that this is the first time that anybody tried to use or validate Granger with biological data from a preparation where you actually knew what the circuit was. So we published the paper, and then we'll never do Granger again. Other people can, but as long as they know what they're doing. Okay. So now I just want to show you one other little sort of excursion into connectomics in this circuit. So the top 2 traces in these 3 panels are just spike times extracted from an extracellular recording. These were extracted from mixed nerves, so Gabrielle just extracted them this way so you can see LP and PD more simply. This is an intracellular recording in each case from the IC neuron, and the bottom 2 are intracellular recordings from LG and DG. So over here, under these control conditions, the pyloric rhythm is on, the gastric rhythm is off, and IC is firing in time with the pyloric rhythm. Then Gabrielle added a little nicotine to the preparation. She turned on LG, DG, so the gastric rhythm turned on, and you can see now a strong modulation of the IC's activity during gastric mill-act time. So during the LG burst, the IC is depolarized. It's firing more spikes per burst. But now we can see simultaneous pyloric and gastric mill activities. Then a little later, as the nicotine really kicked in, the LG and the DG now are very strongly and robustly active. And you can see that IC has now more or less left pyloric time and is firing in small, strong gastric time bursts. So IC is a neuron that switches between firing in time with the pyloric rhythm to firing in time with the pyloric rhythm to multiplexing both rhythms. And so one of the things we've been trying to understand is what are the circuit mechanisms that control neurons that switch between being part of a fast or a slow oscillator or multiplexing them both. And you can see in this demonstration that nicotine is able to gate that. So what Gabrielle did is she looked at this circuit, and then she decided to model or build a model around this sort of central little circuit, this 5-cell circuit. The 2 fast cells in her model are going to be over here. Here's IC, that's the switching multiplexing cell, and 2 slow cells. And so she's now built a cartoon circuit that basically captures this essential circuit to ask the question, what can we understand about how cells might switch for being part of a fast and slow oscillator? So what she's done is she's used now very simple models. These are Morris-Lacar model neurons. They're oscillators. And she set 2 of them up as fast oscillators in isolation. One, the neuron she's calling the hub neuron, or that switching neuron, as as an intermediate oscillator, and the 2 slow ones. Here they are in isolation. When she couples them up in the half centers fire in alternation, as you expect. When she couples them up with the electrical coupling, they synchronize, as you'd expect. And then if she pulls together the whole circuit, depending on conditions, this hub neuron will either be firing in time with the fast oscillator or in time with the slow oscillator. So this guy either couples primarily in with this group or with this group. And now what she can do is she can change the strengths of these synapses or these synapses or these synapses and see what happens to the way the hub neuron is firing. And when she did this, now she's just plotting that hub neuron frequency on a color scale where blue is slow and red is fast. You can see a very interesting set of parameters. What you see is this tongue of high frequency behavior where the IC cell or the hub neuron goes, as you go from zero electrical coupling to higher electrical coupling, goes from being slow to fast into slow. And to really visualize this, or to see this, what Gabrielle did here is if this is A, you're seeing A is fast, fast, slow, fast, slow, slow. Then as she increases the electrical coupling, now it's coupling into the fast, and then it goes back into the slow. So A, B, and C on the graph are shown here. But now this is really cool. I mean, this is why it's really nice to have really creative graduate students, you know, because she did something I never would have thought to do. What Gabrielle wanted to do, and I think this method may be useful for a lot of reasons for a lot of people, she wanted to be able to visualize the frequency of all 5 cells as a function of those 2 parameters. So what she did is she created for each point on this a set of 5 circles and squares, with the outer circle is the fast oscillator on the end. Then the next circle is the frequency of the other fast oscillator, or the one. The square is the frequency of the hub neuron, and then the 2 slower oscillators. So at every point, you can see what's going on to the coordination. So up here, the 4 cells are firing slowly, and only one of the fast oscillators is sort of still going fast. Over here, all 5 cells are going at the same frequency. Over here, you can see you've got one slow cell and the other 4 are going quickly. And you can see at all different regions of what she now calls a parameter scape, you can see what happens with the frequency relationships. And we actually spent some time trying to understand all this, and I don't have time to go through it now, but I want to show you a fundamental take-home lesson for this, which I think is really important for those of you who are using optogenetics or genetic manipulations to try and understand how circuits work. So here's her circuit. Here's a set of starting conditions. That hub neuron, this IC neuron, is firing slowly. These guys are firing quickly. Here, she took these starting parameters and she decreased G syn A. That's the strength of these connections. And you can see the hub neuron now is going quickly. So it goes from slow to fast. Then she went back to these conditions, and now she decreased the electrical coupling strength. And you can see the hub neuron goes to firing into the fast. And here, she decreased these synaptic connections, and again, she produces the same switch. So now you have the same circuit change produced by 3 entirely different circuit mechanisms. Be this a warning, okay? But basically the warning is that it is possible to do an optogenetic or genetic manipulation and see a change and mistakenly assume that it's the answer. It is possibly only one answer, and somebody else in a lab in Tokyo could do a different manipulation and get a very similar result, and you guys can both be right and both be wrong, right? So the important thing is that, similar changes in network performance can arise from a variety of circuit mechanisms. So just like the genetic code is degenerate, changes in circuit function can also be degenerate. Now, I'm going to very quickly bring you to the next step, and I'm going to be skipping a whole bunch of stuff in the middle that I knew I wouldn't have time to do, but to bring you to what we've been doing for the last, most of what we've been doing for the last 6 or 7 years. And this story started with work that Astrid Prince did when she wanted to build a model of the pyloric rhythm. And she wanted first a good model for the LP cell, a good model for the PY cell, and a good model for the PD cell. So she started out with a conductance-based model that had been developed before in the lab, and it had a leak, a hyperpolarization, activated inward current, 3 potassium currents, 2 calcium currents, a sodium current, and all of these had been measured in STG cells using voltage clamp. So she started tuning this model, and she tuned for about a week, and really bad things came out of her mouth. I can't repeat them. I didn't understand them. They were all in German, and when she was cursing in German, you knew it was really bad. My other German postdoc was snorting and snickering, so I knew it was bad. And then, Astrid being very smart, she said, instead of doing all this unhappy work, she decided to let the computer do the work, so she chose 6 values for each of these 8 currents, and she simulated all against all, and so she simulated 1.7 million model neurons, and then she sorted through them to find ones that would be good model neurons for each cell type. And from that, she came up with 5 model pacemaker neurons, 6 PYs, 5 LPs, and then she took the pyloric rhythm architecture and chose either 5 or 6 values for the synaptic conductances, and then she simulated all against all, and so she simulated 20 million model networks. And it ran for a couple months on the cluster. This was early days in 2003. But at the end, she ran all of these, and then she sorted through them and asked, what did she get? And she got what you'd expect. She got some that were triphasic in the right order, like here. She got others that were triphasic in the wrong order. She got others that weren't triphasic and that were really disordered. So if all of them were there, 19% were triphasic in the right order, and 2.4% of them had behaviors that fell within the range of data that Dirk Bucher compiled from 99 animals in which he measured the pyloric rhythms. So he measured the frequencies and the phase relationship, the number of spikes per burst, everything you can imagine measuring from those 99 animals. And then she said she required that these good models, good enough models, fit within that range of the physiologically measured data. So we're not looking for a single model, an optimal model, nothing like that. We're just looking for a population of models that capture the population that we wanted to model. And I actually think this is a much wiser way to go about doing things. Now, 2.4% is a small number, but if it's 2.4% of 20 million, it's 400,000. So she had 400,000 good models, and I'm just going to show you 2 of them. Here were 2 model circuits that were producing very similar motor patterns. And now if you look at their underlying conductances, you see their underlying conductances are quite different. So the AB to LP synapse here is big, here it's small. PY to LP, here it's big, here it's small. The H current in PY is big here, here it's small, etc. So this set of parameters and this set of parameters are completely different, and yet they're both consistent with very similar circuit performance. So when we started talking about this, a lot of biologists loved this. They said, phew, my data probably is really okay, because maybe the fact that I actually see variability is real. I took a lot of trash from some theorists, because some of them really thought that somehow or other the crab should have an optimal single solution, so we won't talk about that. But they made another very important point. We had figured out, we thought, that this kind of difference that you see would be possibly a signature for what kinds of compensations and correlations might be there, and might be very important. And that is true. The other possibility is that some of these big differences are because those parameters just don't much matter, right? And both of those things are true. The other thing the theorist said to us is, okay, it's producing similar behavior here, but because the underlying structures are so different, there will be perturbations that are going to separate these. And that is certainly true. In the universe of all possible perturbations, there is a perturbation that will give different performance from this set of parameters and this set of parameters. So from this paper, we came up with 3 questions. Question number 1 is, how variable are the parameters in real biological networks? Which parameters are highly variable because they just don't matter? And which variations are indicators of network level compensation? And then, how reliable are networks with different underlying structures against environmental perturbations or neuromodulation? So that's what we've been doing since 2004, and today is basically answering these questions. Now I'm going to skip a lot of slides that are in here, so in a moment I'm going to flip through them really fast, and I'm just going to tell you the answers. The answer to this is that synaptic strength and membrane conductance densities vary in the same identified neuron, in the same identified synapse, in our hands and in many other people's hands in the 2 to 6-fold range. So that answer comes from our data, comes from Ron Calabrese's data on identified synapses in Leach, and it comes from many other places. And here's some of that, which I'm going to skip, but it's all published. And if any of you want to see those data, if you do a search on me, you'll find some of it. If you do a search on Ron Calabrese, you'll find the very beautiful Leach work. So this is synaptic and intrinsic membrane conductance vary 2 to 6-fold when measured in the same identified neuron in different animals. Now, that number is far bigger than any of us would have predicted, and it's also not 3 orders of magnitude. So it's a very interesting range, and it's the kind of thing you can imagine is feasible with molecular mechanisms, unlike 0.1%, which would be hard to do with the stochasticity that we all know and love in biological systems. On the other hand, it's a lot bigger than any of us would have thought. But it tells you that the solution space is shallower than one would have thought. Okay. The second question is, the simple answer to the second question is that some parameters, some changes in synaptic strength almost certainly do not much matter. Some synapses only have to be bigger, they have to be big enough, and then if they're 3 times as big, it doesn't matter, especially for inhibitory conductances. And others are definitely very strongly compensated and correlated, and I'm going to have to skip this paper, which was also published. But it basically comes up with the answer that network reliability can be achieved by compensation of variability with variability. And so that is very true. Okay, but I want to spend the last few minutes addressing that last question about perturbations, because it goes to the heart of what I think we all care about. So all of you are healthy. Well, we're all healthy to some degree and not healthy to other degree, but we're all at least healthy enough to be in this room and to have a shared experience, except those of you who are asleep, in which case you're not. And I'm very sympathetic because I sleep through seminars, but you're not supposed to sleep through mine. Anyway, you know, I don't do reciprocity very well. I can sleep, but other people aren't supposed to sleep. Anyway, temperature for a crab or a lobster living out there in the Atlantic Ocean is a perturbation that the crab or the lobster has to be able to deal with, and because otherwise it wouldn't be healthy. Now, I told you that our data shows that our synaptic strengths and our membrane conductances can vary. An LP cell can have a pretty large range in its potassium currents or in its H conductance or in the mRNA for the sodium current, etc., within that range. And the synaptic strengths can vary. So every crab has a different set of solutions. So how do they do with temperature, which is one of the most salient and perturbing perturbations that they see? So temperature, you know, affects every single biological process. We describe it with a Q10. A Q10 of 1 means that a process is completely temperature invariant. I was taught when I was a kid that all biological processes had a Q10 of 2, which is a lie. There's an exponential in here, and so a Q10 of 3 blows up pretty fast. Now, ion channels have Q10s that can be in the 2 to 3 range, or it could be 10. And obviously, some very temperature-sensitive ion channels have Q10s at 50 or 100. So biological systems have a variety of Q10s. And if you think about building a circuit with processes that have a large variety of Q10s, you can say you start with one set of solutions, and now temperature is going to give you a different set of solutions as every one of those temperature-dependent variables changes the effective parameters. So what happens? Well, you all would predict that the pyloric rhythm would speed up as you go to higher temperatures. So Lamont-Tang did these experiments. Here at 7 degrees, we have PD, LP, PY, beautiful pyloric rhythm. The same preparation at 19 degrees, a beautiful pyloric rhythm. And then when he plotted the frequency on a log scale, because that's exponential, as a function of temperature, you get a Q10 of about 2.3. Perfectly normal. However, the remarkable feature is that the phase relationships, that is to say the normalized time at which each cell fires, is completely temperature invariant. So this is remarkable. It says the frequency is very temperature sensitive, but the phase relationships, which you would need for the rhythm to work correctly, is preserved. And we actually understand this pretty well. But to show you how incredibly beautiful this is, these are now intracellular recordings at 7, 11, 15, 19, 23 degrees. And then what Lamont did, nowadays you can do this, he scaled out the time difference and overlaid the traces. And you can see that the membrane potential waveforms that give rise to that invariant phase are almost perfectly maintained. And this occurs because two of the currents that are most important for the phase, IA and IH, in the follower cells actually have very similar Q10s, and that the pacemaker kernel actually turns out to be duty cycle compensated, so the whole thing works so that frequency varies and phase is maintained. Okay, so this is all now at a permissive temperature range. If we go hotter, now we go to the summer, you know? And here we have intracellular recordings. We have a nice pyloric rhythm. Go up to 31 degrees. This crab is now stranded on the beach. And we get what I call a crash. And then you go back to 23 degrees. The rhythm comes back. So this is a crash, not a cook. It's reversible. And now we can ask, how can we characterize the way these crashes occur? And I'm almost done. Promise. So what we've been doing is we've been plotting spectrograms where this is PD, LP, PY, and 0 to 6 hertz, with the power at that frequency shown at high power in red and low in blue. And you can see here at 19, all the cells are firing together in a single rhythm. However, at 23 degrees, you're starting to see disruptions. At 27 degrees, you can see periods of disruption. Here you can see a complete crash over here where everybody became silent. So this allows us just to visualize what the crashes look like. And I will skip that. And now we can just look at 5 animals that were all cold acclimated. And you can see that all 5 of them behaving perfectly well at 15 degrees. They're behaving well at 19 degrees. This one started crashing at 23 degrees, whereas the others look better. And if we look here at 31 degrees, you can see that the forms of the crashes are completely different. They're all showing crashes. They're all showing, to some degree, some amount of intermittent crashes. But here, LP has fallen out. And here you're getting these bouts. And here you're getting fast bouts, etc. This is exactly what you would predict if each of these animals had a different solution. In the permissive range, all of those solutions are good enough for performance. And now you're seeing these slightly different crashes as those different starting parameters are starting to show different dynamics at the most extreme perturbations. And I'm going to skip the next thing and just show you the conclusion from crashes, which is that each individual animal crashes with individual dynamics as predicted by their underlying parameter differences. At low temperatures, all networks are well behaved. showing consistent changes in frequency and outstanding temperature compensation. I didn't show you the data, but temperature acclimation does not reveal any obvious changes in the response to acute temperature in the permissive range, but stretches the range of good enough behavior. The warm acclimated animals don't crash into higher temperatures. I'd just like to end by saying that multiple good enough solutions to circuit performance in the population will ensure a substrate for evolution under diverse environmental conditions. It has to be this way. Each of you has to have a different nervous system, and every crab has to have a different solution to being a crab for evolution to work. And I'll stop there. Bob.
Do you think you would have the same variety of solutions in genetically identical animals?
So people ask me all this. That's why I said it was wild type species. It turns out that if you look in bacteria or in yeast, which are clonal and therefore identical genetically, and you measure the values of any protein or metabolite, they're all over the place. Moreover, bacteria and yeast, not even talking about flies and worms, bacteria and yeast show a tremendous amount of variability in gradients and other kinds of things. And basically, even at that level with a purely genetically identical strain, there's enough stochasticity in every biochemical reaction so that you accrue a different set of conditions, and that sort of just builds up, and that sort of accounts, the accrual of the stochasticity basically ends up giving you more or less the same answer. Now, in genetically identical animals, presumably there will be some restriction on the total range of parameter sets, because we know that there's a higher incidence of schizophrenia in identical twins, but we also know that not all identical twins become schizophrenic, right? So I think it restricts it to some degree, but far less than your intuition says. These animals are 5 or 6 years old. My guess is in a 6-year-old animal, most of this variability is differential history. Yeah?
Yes, in terms of the non-uniqueness of the parameters to the connectome, I think that a couple of observations might be relevant here. First of all, networks are not necessarily uniquely built. There could be redundancy to it. So, part of the
degeneracy, not redundancy.
Or whatever we call it, but there are overlaps. Okay, so in that case, the parts that are somewhat redundant or secondary may not be uniquely identified. There will be a powerful range of parameters comparable with it. The other thing is that neurons in neural networks could be multitasking, could be involved in one behavior at one point and then doing something else totally different, and then could be totally reconfigured with all kinds of neuromodulators and so forth. So it's a matter of the experimental constraints they're under, that will determine the kind of parameters that will be relevant to the situation
and their history.
Well, the history,
Right.
whatever. So that really brings a lot of questions on how to uniquely identify a network, whether there's a unique set of parameters when all of these constraints are taken into consideration as opposed to taking a snapshot of rhythm or whatever input-output relationship at some point, and so on. So,
Well, I think for vertebrate, if you think about cortical circuits, I think that's the right question. In this circuit, that pyloric rhythm is there from when the animal's very, very young throughout its whole life. So maintaining that circuit, just like maintaining your heart function, is absolutely required for the survival of the animal. So you can wander around in parameter space, but there's an imperative to maintain a target activity level in that circuit or in your heart and probably in your respiratory circuits, et cetera. So there's a whole other story about homeostasis and how you maintain a target activity level, which is far harder to think about when you think about things like cortical circuits. But certainly in the spinal cord or in other places, there are some target activity levels which have to be maintained even though the components are changing. Over there. Youth before beauty.
Is there a sense if the solution space is shallow or there's just a bunch of local minima?
We know for the single neuron that where we started that all single spike bursters are in a single continuous dimensional connected space. I suspect that it's shallow and not in separate islands because there's so many parameters. You can wander around in many, if you have lots of parameters, it's much easier to wander around in a very shallow single space. You find disconnected regions much more prevalently when you have many fewer degrees of freedom. So I think there are so many degrees of freedom that I think what's really happening is you have a very shallow base in a very complicated multidimensional manifold, and you stay on it, except when you don't. And that's when you become seizure prone or whatever. Back there?
Yeah, so you showed how different animals have different characteristic crash patterns. Are those crash patterns the same for that animal over time, like short periods of time, or an hour later if you did it again, would it be the same? Or is it just
so anecdotally, so we've tried to do this. Anecdotally, the same animal seems to produce similar crash patterns, which is what you'd predict. We just have technical problems. There are only so many times you can crash it before it really does cook. And, so we don't have, doing the statistics on those patterns is sort of hard to know how to statistically do the right comparisons. So anecdotally, I'd say, I believe, for no statistical reason, I believe that the single animals are far more reproducibly the same than animal to animal, but we just don't have a large enough data set, enough repeated crashes in the individual animals to really do it statistically. But I think if I had the perfect grad student or postdoc who wanted to repeatedly crash these networks and then figure out how to do the right stats, it would be. But it's hard to know how to do the right stats because the crashes are non-stationary. Martha?
Yeah, so it's very interesting because it moves the whole concept of neuroplasticity in big nervous systems to another level. And we've known for a long time that early experience can change the levels of cortisol. People vary in their estrogen levels and things like that. And all of this can actually, this is why these crazy drugs work with it, because they jack something up. They switch you from one thing to another. But to approach that experimentally in a complex system.
Well, some of these early developmental things are really important for establishing your baseline connectome. And then once that is established, that's fine-tuned continuously. But presumably, person one baseline connectome is going to be different from somebody else's baseline connectome at the level of the detail of how many inhibitory projections do you have from this neuron to that neuron, or the actual detail of these large circuits. But all of them in the healthy person are consistent with good enough behavior, and they will probably all crash differently when extremely perturbed.
Well, there's a lot of evidence for that in mammalian systems. People knock out one member of the family, and the other one goes sky high.
Right.
The other one has a slightly different property.
Right.
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 So this is basically
Right. So compensation, and my feeling is that the really interesting knockouts are the ones with relatively little phenotype, because they're right. Well, they're the ones that give you a way into looking for where the compensations and correlations really are, right? Now, not all solutions are possible. You knock out some genes and you get overt disease or death, right? And that's true in our homeostatic models, but there are many other compensations which are really very strong. Raganka?
In your last experiments, in which you varied temperature, and you showed that what the system preserved was the phase relationship. So, in any such homeostasis experiment or demonstration, the idea is that there is a feedback onto some elements in the network or in a neuron that targets the phenomena of homeostasis. So in your case, when you have a phase relationship as a target of homeostasis, what do you imagine is the feedback onto individual neurons that allows this to happen?
So we have to distinguish between two sets of experiments. The acute temperature experiments, when we just took a preparation and just shifted the temperature, that I don't think requires any homeostatic regulation. That just, the constant phase there just arises out of the currents that work to determine phase. So we have the strength and time course of the synaptic inhibition, the amount of IH and IA, and as the inhibition gets stronger, then IH activates sooner, et cetera. So the constant phase sort of is an emergent property of the way those currents work. Now, when we do our 3-week acclimation and we get constant phase, there we have to say that some relationship between the currents that under acute conditions are important for constant phase, some correlated regulation of those currents has to be maintained. Now, the interesting thing about IH and IA is whether you do, in our data, when we just look at correlations between IH and IA in individual cells, there's a very strong correlation between IH and IA. When you do overexpression experiments, there's a very strong correlation between IH and IA. And in vertebrate cells, there's a very strong correlation between properties of IH and IA. So it's possible that there are some, we'll call them hardwired correlations that are critical for certain kinds of compensations that sort of ensure that you always go in the right direction. That said, I don't know. That's the long version to I don't know. But I think some of it is actually built into the molecular correlations that are there. Yeah?
From what I understood, there is a lot of variability measured in like the synaptic conductances and such, but it seemed like the different circuit functions could be reproduced under different connectivity. But has there been...
we don't have different connectivity, right? There's always...
Are there different connectivity?
No. So strengths, so there's always one LP cell. It always inhibits the 2 PD cells. There's always one AB cell. It's always electrically coupled to the 2. The minimal connectome, the backbone connectome, is invariant. What is variable is the number or the synaptic strength, the strength of the electrical coupling, the strength of the inhibitory couplings, and the conductance density of every current that we've ever measured. That said, you know, the expression of which currents each cell expresses or what receptors each cell expresses, I would say that's hardwired genetically in development. So I think development sets up the backbone of the connectome and the backbone molecular configuration of the kinds of ion channels, the kinds of receptors that are put in place. And then fine-tuning is setting the numerical values of those parameters. Now, in cortex, it's harder to know because, you know, how many connections there are between any individual inhibitory neuron and excitatory neuron. You know, that's more like a free parameter. You know, but I think it's really important to distinguish between the things that you do think are going to be genetically determined in a very early in development as part of determining the cell's identity. Of course, in large vertebrate networks, you still have the problem of not knowing about cell identity, but that's, you guys will figure it out. I'll even probably still be alive.
So that's the class speaker, and then you're always